Hello everyone and welcome back. In chapter 20 of Robert Dahlman's Microbiology with Disease and Taxonomy, in this session we will be talking about gram-negative cocci and bacillus. In chapter 19 we talked about gram-positive cocci and bacillus, and chapter 20 we will talk about the gram-negatives, which remember are those two main classes of microorganisms where we had our gram-negatives and we had our gram-positives. Especially when we are in an introductory microbiology class, it's going to kind of be the bulk of what you will discuss and what we've been discussing since chapter 4, very early in the semester. So once again, here are some of the learning objectives. We're going to look at Neisseria in the same way that we did for the gram-positives. We will talk about the structure, the physiology, also the pathophysiology of it, the epidemiology and disease, as well as treatment and prevention. So we'll talk about all those things for the Neisseria. We'll also talk about facultatively anaerobic bacilli, Enterobacteriaceae. We will talk about some aerobic bacilli, Brucellosia, Bordetella, Pseudomonas, Legionella, and Coxiella. So those are some of those things that we'll talk about, and once again, kind of the same format as we had in chapter 19. We'll talk about structure, physiology, pathophysiology, disease and treatment, and so forth. So without further ado, let's go ahead and dive right on in. So gram-negative bacteria are probably the largest group of human bacterial pathogens. And the reason that they can be really good pathogens is because they have that lipid A toxin that's part of their outer membrane. So remember that LPS membrane we talked about? They have a very thin cell wall, but they have this outer membrane to compensate for that. And one of the things that's embedded in that outer membrane is that lipid A toxin. So what that lipid A toxin can do is it can cause a fever, it can cause for the blood vessels to get really large, or what we call vasodilation, inflammation, activating the complement system, shock, which remember is a drastic drop in blood pressure, and it can even cause what's called disseminated intravascular coagulation. And what that basically entails is that the blood can become coagulated within the organ and cause the organ to fail. So most gram-negative bacteria, the way that they're going to get into the body is that they breach the skin or the mucous membranes. A lot of these gram-negative bacteria that we will be talking about, like 37 degrees Celsius, which is human body temperature. And because they have this outer LPS layer, and some of them will also have capsules and other ways to evade phagocytosis, they can also evade other immune systems, evade other aspects of the human immune system. So here we're looking at this chart with potential effects of that lipid A endotoxin on here. So one of the things that it can cause, and let's just pull out the finger. Here we go. Leukocytes can release cytokines. Those cytokines can cause for tumor necrosis factor and interleukin-1 to be released, which can lead to a fever. We can have the loss of plasma, which could reduce the blood pressure and lead to shock. That's because the leukocytes, once they are exposed to this lipid A endotoxin, it releases, the interleukin-1 can release immature neutrophils, and then those neutrophils can damage the blood vessel walls, and because they are phagocytotic, it can damage those walls, and then we can have fever, loss of plasma, which can drastically reduce the blood pressure and go into shock. Interleukin 6 and 8 can also do the same thing. We can have blood clotting reactions, which will lead to this disseminated intravascular coagulation, activation of the complement system, which can lead to inflammation. Inflammation can also lead to shock. So these are just some of the many ways that this lipid A endotoxin can work to cause all of those detrimental effects that we associate with these gram-negative infections. So for the first one that we're talking about are pathogenic gram-negative cocci and Neisseria. Neisseria is the only genus of gram-negative cocci that very commonly causes disease in humans. It's placed in a different kind of class, beta-protobacteria, and it's an phylum called Protobacteria. It's really similar to Bacteria and Acinetobacter bacteria, but it's not exactly the same. It is aerobic, it is non-motile, which means it does not have flagella, and many times this Neisseria is as a diplococci. If we did a biochemical test for it, it would test as oxidase positive, and so that means that the electron transport chain works or operates a little bit differently. Pathogenic strains have these fimbriae on there. Remember the fimbriae, they're going to be structures that allow for attachment. They also have a capsule, which makes it able to evade phagocytosis. And then that Lipid A endotoxin, that cell surface marker that we just talked about in the previous slide, they're also found on this Neisseria. So there are two different species that we know that are pathogenic to humans, Neisseria gonorrhea and Neisseria meningitidis. So here's a lovely picture of the diplococci that we're talking about. There's one, there's there. And we gram stain them. Notice that they're gram staining kind of this red or pinkish color, so gram negatives are going to gram stain red. And then you also are able to see not only the diplococci, but you can see the capsule around them. And then remember that capsule helps them to evade phagocytosis. And any methods that a bacteria can use to evade phagocytosis or any aspects of the immune system will make it more virulent or more likely to cause disease. So going on to Neisseria gonorrhea. Neisseria gonorrhea causes gonorrhea. It is the only found or happens in humans. You're probably very familiar with hearing about Neisseria gonorrhea because it is a sexually transmitted disease. The risk of contracting gonorrhea and that disease setting in is going to increase with sexual encounters. Most cases of gonorrhea that we see are in the U.S. or in adolescents, and then we see the cases that we would find, the epidemiology of it, it kind of starts to decline, or it has been on the decline over the past decades. We find it more common in males than females, and one of the reasons for that is because females can be undiagnosed, or can go a longer period of being undiagnosed simply because of the length of the urethra on there. So some of the issues that we associate with some of the symptoms that we find that are very pronounced in men may not be as pronounced in women, so women may not have be diagnosed as quickly and will not have treatment as quickly. What it can do, now the Neisseria doesn't really attach to the lining of the vagina, instead it kind of infects the cervix and the uterus, and it kind of can hitchhike its way up to the fallopian tubes, and it can hitchhike its way onto the fimbriae, and it can trigger inflammation, fever, abdominal pain, and all of these are characteristics of what we call pelvic inflammatory disease. So if we have chronic infections, they can lead to scarring of the fallopian tubes, and they can also create the perfect storm, or the more likelihood of being ectopic pregnancies, where instead of the egg, once it's been fertilized in the fallopian tube, making its way down and implanting itself into the uterus, it can implant itself into some of that scar tissue of the fallopian tube. So here is a lovely graph that shows the incidences of where we have the highest cases of gonorrhea, and then it also has the incidences and population numbers looking at the decades of it. So as you can see, there's this huge explosion that we had happening between 1970 and about 1980 or 1985, and then we see a drastic decrease in that. One of the reasons we see that drastic decrease at that point in time is because there was sex education was more prevalent to adolescents, and the use of condoms was much more, was talked about more openly, freely. Students had, or adolescents had, more access to condoms and had more understanding about sex education, so that helped to see some of that dramatic decline that we find there. We also see where we have lots of cases happening here, and many of these cases coincide with the availability of sex education for students and the availability of condoms in these different places, and it also has to do with population numbers as well. So the gonococci adhere to the genital, urinary, and digestive tract. They can evade the immune system. They have, the ways they can evade the immune system is that A, they have a capsule around it. Also, they can secrete what are called proteases, or these special proteins that can actually break down the immunoglobulin IgG-A, so immunoglobulin A, or that antibody. And because of the capsule, remember, that's how they can survive within the neutrophils It evades that lysosome vacuole complex. Gonorrhea in men, the inflammation is quite painful with urination, so that's one of the reasons why men are more likely to be diagnosed faster, because that symptom is so pronounced. And then there's this pus-filled discharge. In women, as I said before, a lot of times women can be asymptomatic, but it's left to, if it's prolonged, it can reach its way up to, remember, it doesn't really attach to the cells in the vagina, but it can make its way, it attaches to the cells of the uterus and the cervix, and it can make its way up to the fallopian tubes. And if the treatment, or if a treatment does not happen soon with antibiotics, then it can lead to pelvic inflammatory disease. Infections occur outside of the reproductive tract a lot of times. Sometimes we can't have them happening outside the reproductive tract. If that happens, it can cause pharyngitis, it can cause gingivitis if it's in the oral cavity. And we also see infections of the eye and the respiratory tract of newborns as they're passing through the mother's birth canal, as they pick up the bacteria, as they pick it up as they're going through the birth canal. So as a prophylactic, what happens a lot of times for children, specifically children that are born to mothers that have had very little prenatal care, or to mothers that just don't have a birth plan or don't request this, they will administer erythromycin drops to the babies, and that helps to prevent, or some sort of antibiotic, and that helps to prevent this ophthalmic disease. So they call it neonatal ophthalmia, or blindness, that can be caused from the babies picking up gonorrhea in the birth canal. They used to use silver nitrate, like I was, if you're born in the 70s, you probably had silver nitrate. Or if you're a woman that's had great prenatal care, you're comfortable with your doctor, you can put in your birth plan that you would prefer not to have the antibiotic drops put in your child's eyes, because you know you don't have gonorrhea, and your doctor knows you don't have gonorrhea. But if you're just kind of coming in from off the streets, if you will, and the doctors and nurses have no knowledge of you, you don't have any prenatal care history, they're probably very likely to go ahead, and especially if you don't ask them not to, they're probably very likely going to administer the antibiotic drops. So diagnosis for asymptomatic cases, they can be identified with genetic probes as a way of diagnosing them. We can also look at the pus from the inflamed penis and look and see, as we in gram-thin, and look for these gram-negative diplococci. Treatment can be a little complicated because there are some strains that are resistant. So we like to use broad-spectrum intramuscular, and that's going to increase the dosage amount in the circulatory system. What we do is intramuscular injection of cephalosporins. Prevention is pretty straightforward. Sexual abstinence, monogamy, and proper condom use are all very successful prevention measures. And eye infections are prevented with the use of antimicrobials. And last I checked, I believe it was erythromycin, but I know that in the past, and there may be still some hospitals that are using this, the silver nitrate drops can be just as effective. So going on to our next meningitis, our next Neisseria meningitidis. This is the most common meningitis in individuals under the age of 20. We see babies, infants, toddlers, children that pick this up. It can be caused by the normal microbiota that's in the upper respiratory tract, because some people are just natural carriers of it. It has a polypeptide capsule that is resistant to liptic or lysogenic enzymes that the body makes, and that's what allows it to be evade phagocytosis. Like 30 to 40% of people just normally carry Neisseria meningitis in their respiratory tract, and they're perfectly healthy. When we have crowded conditions, meningococcal meningitis is going to be much more prevalent in young children, and in young adults, especially those adults that are from lower economic groups. So the respiratory droplets, because it can live in your respiratory passages, are transmitted among people living in close contact, specifically families, but think about other groups of population that are in close contact with one another. In the barracks for soldiers, we also have students that are living in dormitories. Those individuals are much more likely, it's the contracting meningococcal meningitis, Neisseria meningitis is much more likely if you're living in closed settings with someone. Also, let's see here, the bacteria, when we talk about that, can be transmitted. Meningitis can cause death within 6 hours if it's not addressed properly. And meningococcal septicemia can also be life-threatening. So the incidence of meningococcal diseases in the U.S. is about 0.3 cases per 100,000 in the population, and most of them usually start kind of abrupt with sore throat, fever, headache, stiff neck, vomiting, and convulsions. For really young children, they may only have fever and vomiting. They necessarily have to have the convulsions. But it can arthritis, partial loss of hearing, and then, you know, getting precipitously worse within 6 hours, it can be fatal. With the meningococcal septicemia, or what we can call blood poisoning, that can be life-threatening because it can trigger shock and blood coagulation in many of the organs, and it can cause these petechiae, which are skin lesions on the body. And then what happens is that because of those skin lesions and because of the hemorrhaging that happens there, you can have these little black lesions that will occur, and it's kind of death of the tissue. And here are just petechiae, that are located on the hands of a patient, 2 different patients, and one looks to be an infant. So diagnosis and treatment. Rapid diagnosis is absolutely critical because the disease can be life-threatening and may cause death within 6 hours. And you may have seen some of the commercials that are advocating vaccination because it can happen so quickly. We look for gram-negative diplococci in the phagocytes of the central nervous system. Treatment, we begin immediately with intravenous penicillin. We start with an IV antibiotic. Prevention, asymptomatic carriers make eradication very unlikely. Because remember, there's like 40% of the population that are just normal, healthy carriers on there. We do have a vaccine against some meningococcal strains, and the CDC recommends that immunization with the vaccine against the strains A, C, and Y, but this vaccine can't be used in children under the age of 2. But we do have vaccinations that are available. So on to the gram-negative facultatively anaerobic bacilli. There are 2 families of that. We have Enterobacteriaceae and Pathogenic. We use an oxidase test to distinguish between these 2 different families. And with that oxidase test, it's going to distinguish whether they're oxidase negative, which doesn't leave kind of that purple color after you use the oxidase reagent on it, from those members that are oxidase positive, which will have a nice purple color to it. These gram-negative facultatively anaerobic bacilli are important in healthcare-associated pathogens, or what we call HIAs, or nosocomial infections. And here's a picture of what will be considered oxidase negative, and then this will be oxidase positive. Remember, the oxidase negatives are going to be the Enterobacteriaceae. And there are about 150 different species, and many of those are important in nosocomial infections. So where we find them is an overview. Intestinal microbiota. It's the intestinal microbiota of many animals and humans. It's found widely in water, soil, and decaying vegetation. Enteric bacteria are the most common gram-negative pathogens that we find in humans. They are motile bacilli and coccobacilli. All of them are going to reduce nitrate to nitrite and ferment glucose, so there would be a positive glucose test, which means it would turn the tube what color? Yellow, right? And then it's also going to have a positive nitrate test, which means when you add reagents A and B on there, you're going to definitely see that color change that happens. Enteric bacteria kind of all have some very similar microscopic appearances and staining properties, so that's why we distinguish them with these biochemical tests that we've been using in lab class. So we look for tests such as motility. We didn't really do motility in lab class, but we do use selective and non-selective media like McConkey's agar or blood agar. We also look for urea utilization, citrate utilization, gelatin hydrolysis, so there's a whole lot of different biochemical tests that we can use to kind of positively identify, because just looking at the structure alone is not going to help much, because many of the different organisms in this family are going to look very similar. So here we have the gram staining take place. So the Enterobacter aerogens is a coccobacillus, and that's this guy over here. And then we have the Escherichia coli, which is this guy over here, which is also gram-negative. And they're both considered enteric bacteria. So just looking at them by themselves is not going to be enough to give you a positive diagnosis just looking at structure alone. And then here's a good picture of dichotomous key that we have been working with, and you should have been working with quite frequently in your lab classes, and that look at remember, the dichotomous key is a series of yes or no questions, and it will bring you to the specific bacteria that you have. That is your unknown. So pathogenesis, members of Enterobacteriaceae have an outer membrane that has that lipopolysaccharide, and it has 3 antigenic components to it. It has a common antigen. It has the O polysaccharide, which has different antigenic properties depending on the species. So we're talking about the family now. And it has that lipid A. So that lipid A endotoxin that we've been talking about, which can cause inflammation, shock, and all a host of other things that we saw there. There are numerous virulence factors, such as they can have capsules that will protect it from phagocytosis, fimbriae, which allow it to attach very tightly to human cells, so it makes phagocytosis very difficult. Exotoxins, which can cause a host of different types of problems or symptoms. One notable thing that it causes, it can cause diarrhea or dysentery, especially if it is found someplace like in the where these enterics like to hang out, which is in your digestive system, in your intestines. Iron binding compounds that will take up iron and make it available to the bacteria, but makes it unavailable to other cells. And then they have what are called type 3 secretin complex, which is a bunch of different polysaccharides that can be made that are made by various different pathogenic enteric species on there. So there are several different ways, there's several different virulence factors that this family of bacteria, these gram-negative anaerobic, facultatively anaerobic bacilli, can utilize to makes it easier for them to set up disease. So diagnosis, treatment, and prevention. The presence of enteric bacteria in the urine, blood, or cerebrospinal fluid is always diagnostic for us. So we culture the membrane. To culture members of these bacteria from clinical specimens, we use E&B agar, which is selected for gram-negatives, and we can also use McConkey's agar to do the same thing. The selected media, as you probably remember, is going to inhibit, doesn't totally prevent the growth of gram-positives, but encourages the growth of gram-negatives, especially E&B. So if it is something that is a very high lactose fermenter, that's usually a very good indication that what we're working with are these Enterobacter in this family here. And then we also do further biochemical tests to rapidly identify the bacteria, and we have some rapid biochemical tests to do that. They don't all have to wait a couple of days. Some happen fairly quickly within a matter of 24 hours. Treatment, if it is diarrhea, is what's being caused here. It's usually self-limited, and the bacteria will be shed off as you release of diarrhea. Internal infection, we use antibiotics to treat it with that, or antimicrobials. And prevention, because many of the times most of these enterics are found in the soil, the water that's been untreated, and we find them in the guts of humans and animals. Proper personal hygiene, good hand washing, and good sewage control to make sure that the water has been treated or sanitized are always excellent ways for prevention. So here we have that McConkey's agar, which was that selective agar that we talked about, and notice that Staph aureus is a gram-positive, E. coli is a gram-negative, so we don't really see a lot of that gram-positive happening there. So that's just a picture to show you how the McConkey's agar can be used to select for gram-negative. And then we also have, not only is it selective, but it's differential, because both the E. coli and the Salmonella are gram-negative, so they both grow, but you have a different picture or different coloration of the Salmonella than you have of the E. coli. So that's where the differential aspect of this agar comes from. It's very similar to mannitol salt agar, where it can be selective for gram-positive, and then it's also differential for different classes or different particular species of gram-positives. It'll have a different coloration to it. And the coloration from the McConkey's agar is whether or not it ferments lactose. So pathogenic Enterobacteriaceae are classified into three groups. They can be coliform, they can be non-coliform, they can be true pathogens. Coliforms are going to rapidly ferment lactose. They're part of your normal microbiota, but can be opportunistic pathogens. Great example of that would be E. coli. Non-coliform opportunists do not ferment lactose. So if we put it on that McConkey's agar plate, then it's going to just give you kind of a yellowish color. It doesn't necessarily give you the nice pink color. And then we have some that are true pathogens. And examples of those that would be considered a true pathogen in this class of bacteria is something like Salmonella. So first, talking about the coliform, they're aerobic and they can be facultatively anaerobic. So if oxygen is available, they're more likely to use that because oxygen is a better way to get energy. We already know they're gram-negative. This whole chapter is about gram-negative. They're rod-shaped. They can ferment lactose because all coliforms are going to ferment lactose. When they do that, they can form a gas. So they would that gas would be captured in that Dunham tube. Very ubiquitous, commonly found in soil, plants, and undecaying vegetation. They can colonize the intestinal tracts of animals and humans. We can find these coliforms in water, which is usually an indication that the water has not been properly treated. Groups of bacteria that are considered coliform, which means that these can be opportunistic pathogens, would be things like E. coli and Klebsiella. E. coli can cause urinary tract infections if it gets out of the gut where it's normally supposed to be in your large intestine. It can also cause horrible bouts of dysentery if it's allowed to overgrow in the digestive tract. So E. coli is probably one of the most important of the coliforms. We use it in lab a whole lot. It's kind of like our little lab pet. E. coli antigens allow us to identify various different strains. Those strains that are virulent have virulent plasmids that they can transfer to one another. These virulent plasmids that have genes that are going to allow for attachment with the membrane, attachment to not only one another, but attachment to other cells, which makes it difficult for the body to deal with it effectively, and also makes it difficult for antibiotics to work properly. Then we have these exotoxins, and with exotoxin, one of the primary symptoms that it causes is diarrhea. It can cause many different types of diseases. Gastroenteritis is probably the most common of these diseases, which is just an infection of your gastrointestinal system. Produces these cramps, diarrhea, nausea, vomiting, and this is often mediated by these exotoxins. It's the major class, or the major cause of pediatric dysentery that we find in developing countries. Now, E. coli 157H7, and we have all those letters that are tacked onto the back of the name because we're talking about the flagella antigens, because it is a motile organism, and we're also talking about that O antigen that's found in the capsule itself. They are probably the most prevalent strain of pathogenic E. coli that we find in developing countries. After it's been ingested, it can start to very rapidly cause disease, and the symptoms that we find are things like bloody diarrhea, severe or fatal hemorrhaging that can take place. It can even cause kidney damage to also happen. E. coli 0517H7 also produces what's called the sugar toxin, and that inhibits protein synthesis to happen within your cells. And we call it the sugar toxin because we first found it in Shigella, which we'll talk about in a second. And what it does is it attaches to the surface of your phagocytotic neutrophils, and then it spreads it throughout the body. And so when that happens, then there's this widespread death of the tissue and the host cells. The use of antimicrobial drugs can prevent that protein synthesis to take place, and it can increase the production of the sugar toxin of the E. coli. So using antibiotics, have to be very careful with that because it can actually promote the bacteria to make more of this sugar toxin. Common cause of healthcare-associated urinary tract infections or nosocomial infections, primarily with the use of catheter. And we just talked about all of these different things with E. coli 157H7. The one thing that we didn't hit on here is that this is also associated with even contaminated or undercooked ground beef or contaminated milk or fruit juice. And for a child that contracts this this bacteria, it can be very fatal for them and very bad for them very quickly. So on to Klebsiella. Klebsiella grows in the digestive and respiratory tracts of humans and animals. They're non-motile, but they do have nice capsules. You'll probably remember doing the capsule stain with Klebsiella. Having that capsule helps to protect it from phagocytosis. It can also become an opportunistic pathogen. The most commonly isolated pathogenic species of Klebsiella is Klebsiella pneumonia, which can cause pneumonia, but it can also be involved in bacteremia, meningitis, wound infections, urinary tract infections. Pneumonia that's caused by this Klebsiella often involves destruction of the alveoli and production of what we call bloody sputum, so you're kind of coughing this up. People that are alcoholic and patients that have a compromised immune system are at much greater risk of this pulmonary or Klebsiella pneumonia, this type of pneumonia, because their body has a very reduced ability to aspirate fluids or oral secretions from the lower respiratory tract. On to Serratia. And notice how E. coli, Serratia, and Klebsiella, these are all bacteria that you've probably worked with, and definitely if you're in my class, but we have certainly worked with in lab class. So you're very familiar with working with them and what some of the results are for their biochemical tests. So Serratia marcans makes a nice, pretty red pigment, but that only happens if we grow it at room temperature. So for some of you all, if I come by and looked at your plates, I'm like, you know, I kind of have a pretty good idea of exactly what one of your organisms may be based on color. So not that you can go off the, you know, and automatically say, oh, this definitely has Serratia, because there's some bacteria that can give you a very similar color. But that nice deep red color, if it's grown at room temperature, it's a very strong diagnostic characteristic of this bacteria. For years, we thought Serratia was totally benign, that it didn't cause any issues at all. In fact, they used to introduce the bacteria in many different types of environment to track how the bacteria usually moves. So by using this method, they were able to follow the movement of Serratia from the mouth into the blood of dental patients, through also finding it in hospital air ducts and whatnot. And they use Serratia marcans in different places to do like kind of I forgot what the technical term is for it, but to kind of, it's like a mock war situation or a mock crisis situation, and they looked at how it could move in the air through San Francisco to mimic how a discharged biological weapon would actually work. Now, as a result of doing this, because they thought it was totally benign, about a dozen people got sick, and one man actually died. But there's still kind of some question about what other infections this individual had. We now know that Serratia can grow on catheters, it can grow in saline solutions, and it can grow in other hospital-associated items, so it is considered a hospital-associated infection. It's more likely to grow and be life-threatening for people that have been immune compromised, so specifically for HIV patients or people that have severe combined immunodeficiency. And we find that it's frequently resistant to antimicrobial drugs. And here is that pretty red colonies that grow at room temperature for Serratia. So other coliform opportunistics, we have Enterobacter, Gonorrhea and Citrobacter. Enterobacter and Citrobacter we have talked about. Like many of the other enterics or the coliforms that we talked about, those that ferment lactose, they're found in soil, water, decaying vegetation, and some sewage. They can hang out in the digestive tracts of humans and in animals. Enterobacter can contaminate dairy products. All three of them can be opportunistic infections, and we see them a lot in patients, once again, like with Serratia, that have a compromised immune system, and we find that they can grow in catheters and in different places like that. Sometimes they're kind of hard to treat because they are, many of them are resistant to a lot of antimicrobial drugs. So now on to non-chloroform opportunistic bacteria. These are going to be the bacteria that are in the Enterobacteriaceae family that do not ferment lactose. So everything that we talked about so far with E. coli, Serratia, Enterobacter, Enterobacter, those were all organisms that could actually ferment lactose. These organisms that we're talking about now cannot. So first we'll talk about Proteus. And Proteus vulgaris, you're probably very familiar with. We talk about that in class. And Proteus mirabilis, we also, that's a possibility to have for one of your unknowns. They're facultative anaerobes. Proteus mirabilis is most commonly associated with human diseases, can cause urinary tract infections, specifically with those patients that have catheters for a very long time. It's gram-negative, obviously. It's rod-shaped. It has a few polar flagella when you put it in a broth media, but they differentiate into just an elongated cell by means of numerous peritrixis flagella when we put it on agar, so it can kind of change the way that it looks one time. When it's in the presence of urea and it's found in the bladder, the microorganisms make a large amount of this urease, so it would give you a nice bright pink color for your urea biochemical test. We looked at that. It's resistant to a lot of antimicrobial drugs. Because of that, healthcare workers must determine proper treatment following a susceptibility test. And researchers have been able to immunize mice against urinary tract infections that are caused by Proteus. Morganella, Providencia, and Edwardsella are other healthcare-associated infections that we see primarily happening in immunocompromised patients. Usually these involve a urinary tract infection. Providencia can cause kidney stones, and we really just see them, this happening in those people that have a compromised immune system. All right, so that's just as good a place as any to end for this session, this lecture, and then we will pick it up with our truly pathogenic Enterobacteriaceae, talking about Salmonella, Shigella, and Neisseria, which can cause the bubonic plague. Ooh, good stuff coming up here. So we'll finish this up pretty soon. Have a great morning, noon, or night.